Dear learners, in this video we are going to study chapter 2 biological classification of class 11th NCERT biology. So let us start. Since the dawn of civilization there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, for shelter and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red bloods and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed, that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic and non-photosynthetic organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organism have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under different systems, the understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. R.H. Whittaker, 1969, proposed a five-kingdom classification. The kingdoms are defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of five kingdoms. So here this is your table 2.1 characteristics of five kingdoms. Here, the characters are cell type, cell wall, nuclear membrane, body organization, and mode of nutrition. In Monera, the cell type is prokaryotic, the cell wall is non-cellulosic, it means polysaccharide plus amino acid, the nuclear membrane is absent, the body organization is cellular, mode of nutrition is by autotrophic, chemosynthetic, and photosynthetic, and heterotrophic, saprophytic, and or parasitic. In Protista, the cell type is eukaryotic, cell wall present in some, nuclear membrane present, body organization cellular, mode of nutrition autotrophic by photosynthetic and heterotrophic mode of nutrition. In fungi, the cell type is eukaryotic, cell wall is present with chitin, nuclear membrane is present, body organization is multicellular or loose tissue. Mode of nutrition is heterotrophic, saprophytic, or parasitic. In Plantae, the cell type is eukaryotic, cell wall is present of cellulose, nuclear membrane is present, body organization tissue or organ, mode of nutrition is autotrophic, photosynthetic. In Animalia, the cell type is eukaryotic, cell wall is absent, nuclear membrane is present, body organization tissue or organ system. Tissue to organ system, and mode of nutrition is heterotrophic, holozoic, or saprophytic, etc. The three-domain system has also been proposed that divide the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain and thereby a six-kingdom classification. You will learn about this system in detail in higher classes. Let us look at this five-kingdom classification to understand the uses and consideration that influence the classification system. Earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperm under plants. The character that unified these whole kingdom was that all organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group, fungi, and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, earlier placed in algae within plants and both having cell walls, with Paramecium and Amoeba, which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristic and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic. It means it is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker's system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separately in chapter 3 and 4. Kingdom Monera. Bacteria are sole members of kingdom Monera. They are most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans, where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrium, and the spiral-shaped spirillum. The plural of coccus is cocci, the plural of bacillus is bacilli, and that of comma-shaped vibrium is vibrio, and that of spirillum is spirilia. Here, this is cocci, spherical shaped, this is bacilli, rod shaped, this is spirilla, spiral shaped, and this is vibrio, comma shaped. So the bacterial structure is very simple. They are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic. It means they synthesize their own food from inorganic substances. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs. It means they depend on other organisms or on dead organic matter or food for food. 2.1.1 heading is Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extremely salty areas, halophiles, hot spring thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. Next heading 2.1.2, that is eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by presence of a rigid cell wall and mobile flagellum. The cyanobacteria, also referred as blue-green algae, have chlorophyll as A similar to green plant and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous, freshwater or marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They are often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, example nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria. These are the most abundant in nature. The majority of are important decomposers. Many of them have significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Figures 2.3. Here it is. Figure 2.3, dividing bacteria. This is the cell wall. This is the cell wall. This is the inner membrane, that is called cell membrane, and that is DNA. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they reproduce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animal and plants. Kingdom Protista. All single-celled eukaryote are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protista to one biologist may be a plant to another. In this book, we include chrysophyte, dinoflagellate, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. These kingdom forms a link with others dealing with plants and animals and fungi. Being eukaryote, the protista cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Chrysophytes 2.2.1. These groups include diatoms and golden algae, desmids. They are found in freshwater as well as in marine environments. They are microscopic and float passively in water current. Plankton. Most of them are photosynthetic. In diatoms, their cell walls form two thin overlapping cells which fit together as in soap box. The walls are embedded with silica and thus walls are indestructible. Thus, diatoms have left behind a large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the oceans. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigments present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and other transversely in a furrow between the wall plates. Very often red dinoflagellates, example Goniolax, undergo such a rapid multiplication that they make a sea appear red. Red tides. Toxins released by such a large number may even kill other marine animals such as fishes. Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein-rich layer called pellicel, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoid are identical to those present in the higher plants. Example, euglena. Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic material. Under suitable condition, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable condition, the plasmodium differentiates and forms fruiting bodies, bearing spores at their tips. The spores possess true walls. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoan. These organisms live in freshwater, seawater or moist soil. They move and capture their prey by putting out pseudopodia, false feet as in amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Flagellated protozoans. The members of these group are either free-living or parasitic. They have flagella. The parasitic forms cause disease such as sleeping sickness. Example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms. Because of the presence of thousands of cilia, they have cavity gullet that opens to the outside of cell surface. The coordinated movement of rows of cilia causes the water laden with food to be steered into the gullet. For example, paramecium. Figure 2.4d. Figure 2.4d. Figure 2.4, where is this? Here. This is paramecium. Okay. Sporozoan. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasites which causes malaria, disease which has a staggering effect on human population. Kingdom fungi. Fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaves are due to the parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the sources of antibiotics, example penicillium. Fungi are cosmopolitan and occur in air, water, soil and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long cylinder thread-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called cenocytic hyphae. Others have septae or cross walls in their hyphae. The cell wall of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depend on living plants and animals are called parasitic. They can also live as symbionts and association with algae as lichens and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission and budding. Asexual reproduction is by spores called conidia or sporangiospore or zoospore, and sexual reproduction is by oospore, ascospore and basidiospore. The various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves the following 3 steps. First, fusion of protoplasm between 2 motile or non-motile gametes called plasmogamy. Second, fusion of 2 nuclei called karyogamy. Third, meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, 2 haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of 2 haploid cells immediately result in diploid cells. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage N plus H in 2 nuclei per cell occurs. Such a condition is called dikaryon and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi form fruiting bodies in which reproduction division occurs, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation and fruiting bodies form the basis for the division of the kingdom into various classes. Phycomycetes 2.3.1, that is a type of fungi. Member of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places, or as obligate parasites on plants. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospores, motile, or by aplanospore, non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of 2 gametes. These gametes are similar in morphology, isogamous, or dissimilar, anisogamous or oogamous. Some common examples are mucor, rhizopus, the bread mold mentioned earlier, the albugo parasitic fungi on mustard. Ascomycetes, commonly known as sac fungi, the Ascomycetes are mostly multicellular, penicillium or rarely unicellular. Example, yeast. Saccharomyces, they are saprophytic decomposers, parasitic or coprophilous, growing on dung. Mycelium is branched and septate. The sexual spores are conidia, produced exogenously in a special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores. which are produced endogenously in sac like asci, singular ascus. These asci are arranged in different type of fruiting bodies called ascocarps. Some examples are aspergillus, claviceps and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicacies. Basidiomycetes. Commonly known as forms of basidiomycetes are mushrooms, bracket fungi or puffballs. They grow in soil and or knocks and tree stumps and living plant bodies as parasites. Example, rust and smuts. The mycelium is branched and septate. The sexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis takes place in the basidium, producing 4 basidiospores. The basidiospores are exogenously produced on the basidium. Plural is basidia. The basidia are arranged in floating bodies called basidiocarps. Some common members are agaricus, mushroom, figure 2.5c, astilago, and puccinia, rust fungus. So, figure 2.5c, agaricus, that is a mushroom. Deuteromycetes, that is the last one, type of fungus, commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given a name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Coleotricum, and Trichoderma. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organism commonly called plants. A few members are partially heterotrophic, such as the insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plant includes algae, bryophytes, pteridophytes, gymnosperm and geosperm. Life cycle of plants has 2 distinct phases, the diploid saprophytic and the haploid gametophytic, that alternate with each other. The lengths of the haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups in plants. These phenomena is called alternation of generations. You will study further details of this kingdom in chapter 3. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell lack cell walls. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves and glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female, followed by embryological development. Saline features of various phyla are described in chapter 4. Viruses, viroids, prions and lichens. In 5 kingdom classification of Whitaker, there is no mention of lichens and some cellular organisms like viruses, viroids and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effect viruses can have on us. Even if we do not associate it with our condition, viruses did not find a place in classification, so they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of host cell to replicate themselves, killing the host. Would you call the viruses living or non-living? Viruses means venom or poisonous fuel. Dmitry Ivanovsky in 1892 recognized a certain microbe as casual organism of mosaic disease of tobacco. These were found to be smaller than bacteria because they passed through bacteria-proof filters. M.W. Bazering in 1898 demonstrated that the extract of infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus and called fluid as Contagium vivum fluidum, infectious living fluid. W.M. Stanley in 1935 showed that viruses could be crystallized and crystals consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to protein, viruses also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infection. In general, viruses that infect plants have single-stranded RNA and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double-stranded DNA viruses. The protein coat called capsid made up of small subunits called capsomers protects the nucleic acid. These capsomers are arranged in helical or polyhedral geometric forms. Viruses cause disease like mumps, smallpox, herpes and influenza. AIDS in humans is also caused by virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viruses in 1971, T.O. Diener discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight, prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable disease caused by prions are bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and its analogous variant Creutzfeldt's Jakob disease, CJD, in humans. Lichens are symbiotic associations. It means mutually useful associations between algae and fungi. The algal component is known as phycobiont and fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrient and water for its partner. So close is their association that if one saw lichen in nature, one would never imagine that they had two different organisms within them. Lichen are very good pollution indicator. They do not grow in polluted areas. So here the chapter is almost finished, so summary is remaining. Okay, so here the chapter is finished. Bye bye.